So in this first code chunk, I give you instructions here. Make the answers data frame for section 4. Call it section 4. Write your code below. So you look back at assignment A, find the code and change it as needed for you to be able to create the data frame where you're going to save your answers for this section that you can submit. Okay, so we start here with the resampling heading. There I say, in reality, we can never do 1,000 different experiments on 1,000 different samples from our population of interest. And this is where resampling is valuable. So again, you've probably touched on this in your lectures by now. But as we spoke before, if you imagine this big population that we are interested in, whatever your scientific question is. So we do a lot of TB studies. If we are interested in some TB question, then essentially our population, our study population that we care about, is all TB patients. And sometimes our questions are more specific, so we may say all TB patients in high prevalence settings. Because like South Africa, we're a high prevalence setting, so that means we have lots of TB and a high community exposure, especially in some communities. And if you compare that to a country like Norway or Sweden, they do have TB cases, but mostly their TB cases are from people who immigrated to those countries or are asylum seekers, or they came from high prevalence countries and are now staying in those, like in Norway or whatever. So they don't have anything like the TB epidemic that we deal with in South Africa. And many times that makes the cases that they see so different, actually, that sometimes the kinds of results that we get in South Africa, for example, are actually not applicable to Norway because their epidemic looks so different. So we might sort of narrow down our study population a little bit, like I said, to say, yes, we are interested in TB, but specifically in TB in high prevalence settings, because sometimes the results we get in a high prevalence setting isn't necessarily generalizable to a country like Norway. So that's very important at the start of a study, to understand who is your study population. And it doesn't have to be humans. So if you are doing mouse experiments or whatever, or if you are doing experiments on populations of cells, for example, then maybe those cells are your population. But you have to have a very clear idea in your mind what is the point of doing your experiment? Where do you want to generalize your results? So if you are using, like a cell culture or whatever, as your population, let's say you're using lymphocytes or something, then you have to have a clear idea in your head. If you get a certain type of result in a laboratory-based cell culture of lymphocytes, how can you generalize those results? So probably you want to say something about what lymphocytes in humans are doing. If you're in our environment and you're studying TB, or maybe if you're in the animal group, maybe you want to generalize it to a certain animal, but you're obviously aware of the limitations of that cells do not behave the same in a laboratory, in a culture, in vitro, as they would behave in vivo inside a person, surrounded by all sorts of other cells as well, and all the exposures that a person is exposed to. So you have to be aware of who is the population that you want to generalize to. And then once we know who the population is that we want to study, the very best way to do it, as we said before, we can't study everyone in the population. So the next best alternative is to do just a whole bunch of experiments on the same population. And so, in a sense, we are sort of doing that because we are thousands of scientists all over the world doing experiments on TB patients. And so, in a sense, we are doing thousands of experiments, but we're not doing the same experiment. So, the study that I'm doing isn't exactly the same as a study at UCT who sort of asks the same question, but maybe they have different criteria for which age groups they're including and things like that. So it's not exactly the same experiment. So the next ideal situation would be if I myself could do thousands of experiments that are exactly the same, but I just take thousands of different samples from the same population, and eventually I can put all those results together. Even that is not practically possible. And like I say, even though we are thousands of scientists doing thousands of experiments, it's still not, our experiments are not similar enough to just simply combine the results and interpret it like that. So this is where resampling statistics came up with an interesting solution, saying, okay, well, we don't have the whole population and we don't have thousands of unique samples, but we have this one sample. So like in our case, each of you has a data set of 400 patients. So since we have 400 patients, let's see what we can do with 400 patients. And so the idea of this resampling statistics is it's creating this artificial population almost. So it's saying, okay, it's almost like saying, let's play a game. Let's imagine these 400 patients, providing they are representative of the population that we actually care about, then let's pretend they are not unique individuals, actually. Let's pretend we can sample them over and over again and just create different combinations of them, as if they're actually not just these 400 individuals. We are basically creating artificially a much bigger population by just using different combinations of the 400 patients we have. And so also, as you can imagine, either for us to create different combinations of these 400 patients, we have two options. Either we have to take smaller groups. So we have to say, okay, we're not going to use all 400 at a time because there is only one combination of 400 people. Once you have 400, you can't shuffle them or anything, So either we'll have to take smaller groups, so we'll have to take 100 at a time. If you think back to putting, it's as if you put every patient's number in a bag and put your hand in and draw out samples. If you're going to draw out 400 samples and you're not going to throw them back, you're going to draw them out and leave them out, then you can only draw 400 and you only going to draw the same 400 you already have. So that doesn't create anything artificial or anything. That's just your sample. So either you have to take smaller groups, and so you draw out 100 at a time, for example, and throw them back, and then draw out another 100 and throw them back, and that's the only way you're going to get different combinations, and you can keep on doing that. You're not just going to do it 4 times because then you've reached 400. You'll keep on doing that. Let's say you do it 100 times. Because there are many different, you are going to get unique, most probably going to get 100 unique combinations of patients if you throw them back, draw out 100 again, keep on doing that. So either you're going to do that, or you can do this interesting thing which we call the bootstrap, which is to sample with the replacement. So like I explained before, if you draw from a bag the numbers and you don't put the numbers back, a number back before you draw one again, then that's sampling without replacement. So the patient that's already been drawn can only be drawn once, and so he cannot be drawn again. But if you do sampling with replacement, that means you take out a number and you throw it back immediately before you take out the next one. And so that patient has a chance to be picked more than once. And so obviously in real life, that is strange, and you wouldn't, you couldn't pick the same soccer player twice for your soccer team. So you have to have 11 players, and you can't have one player can't play in two places at the same time. So in real life, that doesn't work, but that is what we do in resampling statistics. It's as if we're duplicating some of the patients. So if I throw back every number after I drew it out, and then I put in my hand again, some of them will be picked twice. And so in that case, if I take out 400 people for my sample, for a new sample that I want to take, some of those people will be picked twice. And then if I do that 1,000 times or 100 times, take samples of 400, but in this way, sampling with replacement, then I can get unique sets of 400 by sampling from the same 400 I already have. But because some people are picked twice, I end up with unique combinations. So I can take out many samples like that, and in that way I'm creating this artificial population, which I don't have in real life, but in our artificial population we can do this because we're using some people twice. And there are obviously limitations with this approach, but there are also limitations with having only 400 patients. So it is one way to overcome some of our limitations with having only 400 patients, is creating this artificial population. If you have very few patients, if you have only 10 patients, then it's probably resampling statistics is probably not for you. It's not, that doesn't make sense to have only 10 patients and now you're just going to keep on creating like a little artificial population. 10 patients is probably not enough to do this with. But if you have, like us, 400 patients, then it's been shown, and that's why resampling statistics exist, it's been shown that it isn't a bad approach. And so that's basically the principle of what we are going to do in this section now.